Thanks, Nancy, and thanks for the organizers for the opportunity to be here. Everyone's made comments about how warm, quote, it's here. The reason is you sent all the cold weather down to where I live, and right now we have had the coldest temperature in 35 years, and there were literally tens of thousands of broken pipes in the city, so plumbers are having a field day. And in my own building, all our deionized water system is off because the pump froze. But this is a topic that I really developed an interest in while teaching cytogenetics, and then I've had an opportunity to develop it more on the various ILSI task forces. And I got asked to talk about the molecular biology of plant genomic changes, and I guess the reason is because what could be the role in unintended effects. So we're all here, on the premise, is engineering safe? And I want to begin with a quote, and I think it's easy to find any number of quotes similar to this one. We have recently advanced our knowledge of genetics to the point we can manipulate life in a way never intended by nature. Therefore, we must proceed with utmost caution in the application of this newfound knowledge. What makes this particular quote stand out from other similar ones is that it was uttered by Luther Burbank over 100 years ago. So the more things change, I think the more they stay the same. Or perhaps another spin on it would be that we continuously overestimate how harmful genetics can be. So what's the concern? We've all talked about, we've talked about the things that can happen, but not necessarily the why. And I think one of the clearest articulations as to why we're concerned came from this paper in Science by some of the people at FDA. And they mentioned then that plants have metabolic pathways that no longer function because of mutations that occurred during evolution. And that products or intermediates of some of these pathways could include toxicants. And such silent pathways may be activated by the introduction or rearrangement of regulatory elements and activation of repressor genes, point mutations like Mark was talking about, insertional mutations, or chromosomal rearrangements. So this impacts the way that people choose a lead event to put forth for commercialization. You try and make sure there's no flanking genes whose regulation might be changed by the transgene. Try to avoid, to the extent possible, filler sequences of vector backbone DNA. Point mutations, now called SNPs, you try to avoid those. And ensure that fertility is not affected. Normally, chromosomal rearrangements will affect fertility. So the question then is, beginning with, what do we know about insertional mutations? So the first question is, can an insertion really activate a pathway? And the answer is a very clear yes, it can. And perhaps the most well-known example of this is yellow corn. And corn is supposed to be white, like rice. But what happens is there's a transposon, just 382 insertion that inserted itself somewhere into the promoter of the phytoin synthase, and it allows the carotenoid synthesis pathway to be expressed in the grain, and leading to zeaxanthin and other carotenoids being made there. So a perfect example here. The main cause of insertions, as Phil alluded to earlier, are transposons. And there are different types of DNA that can move naturally around the genome. And I think everybody is familiar with the phenotype that a transposon can give. You've seen the striped variegated flowers. The whole premise is that, or the way we used to see transposons, is that you had a gene, transposon jumped into the gene and therefore kept that gene from functioning. So if we had a gene for anthocyanin synthesis, you disrupt it, there's no pigment formed, you get white petals. So if you look at these as maybe the primordial cells that are going to divide to give rise to this flower, and the gene, the transposon, jumps back out of the cell it jumped into, pigment formation is restored, and then as that flower grows, every cell that divides from that original one is going to show this pigment, and that's why you get these pie-shaped slices. And, corn is probably one of the better examples of where we know that there's all sorts of transposon types, and they have very, many times even attractive phenotypes, and they've been part of the diet pretty much forever. And other than that, so they're actually a major component of plant genomes. Every single plant genome ever studied has transposons of various types. Here's just one of the first examples. It's just a very short piece of a chromosome from B73. It's a corn inbred. It's one of the 2 parents that are used in most commercial inbreds. In this little piece, you have 6 different insertions into it. And if you look at the same segment from another corn inbred, you have a whole different set of transposon inserts into it. So there are other examples then of insertion-activated pathways, and we can begin with oranges. Oranges are supposed to be orange, and unless they have an insertion, and then you can get anthocyanin production where it's normally not found. And upon excision of the, or remove, when this goes away, it has altered the structure at the promoter region, and it leads to even more expression of it. And then here's another orange where a similar transposon independently inserted again, slightly different position, and a very similar appearance results. Grapes are a wonderful example. For all of you that enjoy a good Cabernet, really the only difference between a Cab and a Chardonnay is a transposon. In this case, the transposon stopped the pigment production in the skin. But if you didn't know that Cabernet grapes existed and Chardonnays were the only ones to be there, upon excision of the transposon, you partially restore pigment production. So, just because this has no pigment doesn't mean the capacity for pigment production is not there. So the safety issue is that in all these cases I just showed you, the color change comes from a pathway expression in novel tissue. And the way I have an example of carrots here, carrots are supposed to be white, and about 300 years ago they found the orange mutant where all our orange carrots from, and to this day I don't think they have found a mutation that enables carrots to be orange. But we've been eating them for 300 years. But back to the issue at hand is what instead of a pigment that had a color, we had activated something that was colorless, that was tasteless, and toxic. What are the consequences? That's sort of the concern that Kessler et al. Had in science. So to start trying to address this, I ask, or at least 2 questions. One is, how common are genomic rearrangements? And Phil alluded to that they're very common. And how often do they activate production of novel toxins? And I want to begin with just a brief review of some of the genomic changes. And I'll begin with this one. This is a pedigree of different soybeans, 30 different soybeans. And as with any pedigree, the closer the branches are, the more closely related they are to each other, to where you have 2 sister varieties here. And the thing to pay attention to are these purple or pink numbers. And they represent the number of insertions that are found in that one soybean and not in the other soybeans looked at relative to the reference genome. And this 30 soybeans have over 25,000 unique insertions not found anywhere else. And even within the 2 sisters, there's 527 insertions that are only in one or in the other. In an earlier example, a colleague down the hall from me in Georgia was doing this work, and that's MPing is a type of transposon found in rice. And they were looking at an ancestral Japanese variety, and there they found, on average, say 50 insertions per plant per generation. Okay? Some of the newer varieties, I think the breeders have managed to stabilize the phenotypes, and they're talking about, every generation, 1 out of 3 plants is going to have a new insertion. But again, the point of the matter is, a quote from that these were things that go on in farmers' fields day in and day out. Elongated fruit in tomato. This is a mutation that was found in Spain. So it probably happened in Spain sometime after a tomato got there. But what you have is there's this 25,000 base pair region of chromosome 10 in tomato, and that somehow got copied and inserted into this gene that's on chromosome 7, such that now the promoter of this gene is driving the sun gene, which is involved in auxin synthesis, and the result is long fruits. And even the large beefsteak tomatoes are also the result of insertions. So here's the wild tomato, and here's a cultivated one, and this one is basically a larger version of this one. It's got its 4 locules, whereas here the locule number has greatly increased. And there is a transcription factor then, and it's got a small insertion, 6 to 8, actually it's not that small, 6 to 8,000 base pair insertion in one of the introns right there. And that then disrupts it, and the number of locules keeps on dividing, and you get large tomato. Organelle, this is a beautiful example of a chromosome painting. Each column is one of the 10 chromosomes of corn, and the rows are different inbreds of corn. And this happens to be, they took mitochondrial DNA and hybridized it to the corn, and this is what those arrows are, indicating where mitochondrial DNA has inserted itself. And pretty much anytime you find unique insertions there, the thought is they've happened since people started making the corn inbreds. Corn inbreeding started in the 20s, 30s, 1940s, around that type. And there's a similar one for chloroplast DNA that I'm not showing you in the interest of time. But not all inserts have to come from the plant itself. They're sequencing the peanut genome down the hall from me, and they were really excited because they found a lot of Pararetroviruses in the genome, and that reminded me of this work. This is banana, and this is in situ hybridization, and these red arrows indicate virus genomes that have been inserted into the chromosomes. And here you see the same thing in petunia. These red dots are where the virus has inserted itself. This is a type of virus was discovered when they started micropropagating banana, and that they put in clean, non-virus infected plants into tissue culture, and they were getting virus infected plants out, and that really amazed people. And it turns out that this virus can integrate, and how long it's integrated into the genome, it probably can be a few days, or thought to be thousands of years, and then it decides to jump back out, and it causes an infection. Sometimes it does not jump back out, and it accumulates enough mutations that it's stuck there forever. So, for example, the tropical rices have 74 viral inserts, and the japonica, the temperate rices, the short grains, have 88 inserts in them. And the tomatoes, again, these red dots are all one pararetroviral insertion. So the other concern is point mutations. Just to make sure we're on the same page, by a point mutation, a SNP is when one base pair is changed to another one, and an indel is where DNA is present in one individual and absent in the other, and we don't know if this one got inserted here or if this one got deleted there. And they happen all the time. This is a paper that just came out. I guess technically it's supposed to come out in March. But the point of the matter is that you can do paternity testing on identical twins now and decide who was the father, something that could not be done previously because of SNPs. The measurement of new SNP creation has been measured in Arabidopsis. It's about 7 SNPs per billion base pairs per generation, which translates into about 175 SNPs for every 100 Arabidopsis plants. And there's a study out of corn that if you take one protein from 2 different inbreds of corn, say this protein is somewhere between 300 and 400 amino acids long, it can be expected to have 3 to 4 amino acid changes simply due to SNPs. And they're all over the place. Looking at 103 lines of corn, they have cataloged 55 million SNPs. The soybean people, we're way behind. We only have 5 million SNPs cataloged. And we only use about 50,000 for breeding. But as far as impact on plants and phenotypes, here's a modern picture of corn, the type of wheat, the type of wheat that we would all be familiar with. But it wasn't always so. Here's a picture of taken right after the Civil War in the United States. Look how tall the wheat was, right up to his shoulders. Here's another one out of Holland, Bruegel's wheat harvesters. And again, look how tall the wheat was. Whereas, old wheat and new wheat, and the difference is SNPs. There's 2 transcription factors, each have a SNP in them, and that is what gives the shorter phenotype. Or even things like shattering. Our grains, we can harvest them because the seeds do not fall off if the wind blows or an animal walks through them. And again, the ability to retain the grain on the head is a SNP. But then there's other sorts of changes that happen besides SNPs and insertions, and that also have affected plants. Probably the biggest surprise to all is still the idea of presence-absence variation. The idea that every individual of a species had the same genes is out the window. The corn people are still trying to catalog how many genes are different between the 2 major inbreds they use, and it's in the thousands. But one work looking at the sequence, 2 genome sequences, 2 genotypes of potato, 275 genes different. Wild versus domesticated soybean, 856 genes. Just 4 varieties of soybean, 133 genes that are in one variety and not the other. This is an interesting story. It hasn't been published yet, but Michelle Graham from Iowa lent it to me. It's a resistance gene to Asian rust. Here's a soybean leaf. It's at the early stage of a rust infection, and pretty soon that leaf will be dead. But at the locus, there's 8 different genes there. But if you look at all the susceptible types, there's only 3 genes. In another case is copy number variation. This one happens to be to the root knot nematode. And these are 2 root systems. This one's been destroyed by root knot nematodes. This is healthy. And whether it's resistant or susceptible boils down to this 3-gene region here in the genome. And here's an in situ of a susceptible showing this red probe. It's present in 1 copy. But one resistant genotype has 3 copies, and another resistant one has 10 copies, all in a row. Other things like height instability in sorghum. Anyone ever driven by a sorghum field, there's always these that revert to the wild type height. And it turns out that the short ones have they're in one of their, auxin transporter genes, there's a duplication that has taken place in the final exon of the gene. And it reverts back to the wild type, and when it reverts back to wild type, you get the tall sorghum. And you can see the number of tall heads in a field. This happens at a fairly high frequency. One of the more exciting ones is the submergence-tolerant rice. When a rice field floods, the rice drowns normally. And what you see in this plot is regular rice and the submergent-tolerant rice in the back. And regular rice has a gene, it's an ethylene response factor, but the sub-1 one hasn't, this part of the gene here has been duplicated, in this one, and the result then is a pretty dramatic phenotype there. And then there are other ones. Everyone's familiar with the commercial soybean, which is buff in color. And color in soybean is normally just two clusters of chalcone synthase, each one having 3 genes. And apparently, somewhere in here, we get double-stranded RNA and we get gene silencing. But anyone knows when you harvest soybean, you tend to find a few black ones. And that is due to a deletion of this entire cluster over here. Some of the red corn stalks, for example, the chalcone synthase gene is present in one copy, and in the green ones, there's been a duplication event. You can get a hairpin here, get the whole silencing mechanism going. The low glutenin rise, it says, for people that need to restrict their protein intake, there's a mutant rise that's naturally low in proteins. If you look at the genomic locus, you have two, a head-to-head version, I guess it's tail-to-tail versions of the glutenin genes. And whenever there's been a deletion of the DNA between them, they're close enough, again, that you kick in the RNA silencing pathway. And then there's what breeders have done. We actually had a time when people were not afraid of genetic changes. Like, this is a really cool ad from Popular Mechanics back in 1961. Science Bargains, the Atomic Gardening Adventure, because they were going to give you things that were completely unpredictable. And unlike anything yet known, just what regulators love to see in dossiers. And the thing about it is, look at the price, $3.95. That is $30 US in today's prices for 4 seeds. So, the point of the matter, though, is that it is a breeding technique. And this is a facility in Japan. You have the cobalt-60 source in the middle. You have the plants being irradiated around it. And the International Energy Atomic Agency happens to list over 2,500 known varieties developed from such breeding. And you're familiar with a lot of them. For example, some of the pink grapefruit are from mutagenesis. A lot of the string beans. And I think pretty much all the pasta wheat has been irradiated at some point in its pedigree. And the vast majority of the lesions have never been characterized. And I did a literature review trying to find out what was going on. Most of the work came from Arabidopsis. A few of it came into high oleic soybean mutants. But, there's always deletions from really tiny of 4 base pairs all the way to 8,000 base pairs. Large inversions, insertions, frame shift mutations, premature stop codons caused by SNPs, among others, are the types of things you see. So the bottom line is that at the end of the day, crops are genetically unstable. And I found this particular paper I thought was appropriate for this occasion at this particular place. And the thing is that the reason we don't have to deal with this on the farm is because we have this huge seed certification programs in place. Because cultivars were unstable, they went through certified seed. The original seed stock is planted, all the off types are pulled out, the rest are let to, you harvest the seed, you repeat the process, and the seed goes on to the farmer. So we purify our varieties to keep the instabilities from accumulating. But a lot of them are, biologically, they're the type of things I just showed you, but a lot of them are due to stress. Here, look at this one. Virus infection promotes genetic rearrangements in systemic Arabidopsis tissue. And it's a good Canadian paper. Another one, Canadian paper, tobacco emits volatiles that trigger genomic instability in neighboring plants. It's par for the course. So it gets us to the last question. Is all these changes that go on, how often do they activate the production of novel toxins? Well, again, I spent many days doing literature reviews. And there are reports where crops have turned toxic. Individual fruits within a variety have turned, of either squash and zucchini. We got, there's 2 really widely grown cultivars in Australia, and a few individuals one year were toxic. They were very bitter, but the people ate them anyway and got sick, and probably well deserved. But another report in California and Alabama. So this is probably some sort of a genetic instability going back to original toxin production of known toxins. Celery is often mentioned, but what really happened there was the Gulf War. Because of that, the celery could not be harvested, and when they went in, it was much later than normal, and at which point it's normally toxic anyway. And there's this case of this potato in Sweden. It was the third most widely grown potato for 50 years, and in 1986 it turned bitter and turned gastric distress. So I don't really consider these to be due to breeding, but due to something else. The question is, what toxic varieties have been bred and become unintentionally toxic? And I think at least 2 of them have. So of all the millions of conventional varieties that have come out there probably the past 100 years, 2 have been reported. There was a potato that had people eating it would have resulted in stomach aches. And then there was a celery that gave dermatitis. And this one, it was the workers in the supermarket that were putting it on the shelf. And then, even then, the only reason they had an issue is because they went to a tanning parlor. So it was a combination of all those events. But all of these have, the previous ones and this ones have all involved elevated levels of very well-known toxins. They're on the OECD list. And it's sort of par for the course. If you're working on a plant that has a known toxin, then you test it to make sure you haven't inadvertently raised the levels of that toxin. What about unknown toxins? In all the history of breeding, I was not able to find a single report that reported a brand new toxin coming into existence. Not one. The caveat to that is there is one example in some of the EFSA documents that they use to justify that, and that is a potato somatic hybrid. They say Lorella et al. 1986 did protoplast fusion, and they got demycin, a new toxin there. But the problem I have with that is that a very exact toxin had already been reported in potato back in 1981. They just did a lousy literature review. So I get back to my premise that in all the history of breeding, I have yet to find a single case of a novel unknown toxin appearing. So, at the end, GMO is just much less viable than breeding. This is genomic comparative hybridization. What it basically is you hybridize the DNA from 2 individuals. And as long as the DNA from one parent is identical to the DNA from the other parent, you're going to get a straight line and background noise. This one is compared to one that was shot with fast neutrons. And the fast neutron deleted 6,000 base pairs. So now there's a region where the DNA is not the same, and you detect it with this thing coming down here. Compare that, though, to just 2 different varieties of soybean, and you get this. Every time you see a peak, it's DNA that's in one that is not in the other. And you can compare it to the GMO version of the same thing, and you really don't detect anything above background. So it gets that, if this does not produce unintended changes that are hazardous, I don't think this should be any more likely to produce them than traditional breeding should. So in conclusion, the changes that cause concern about transgenics are really common in conventional crops, and they affect agronomic and horticultural crops, not innocuity. And we have just picked out to examine the ones that caused a change that was desirable. There's probably a lot of them create no change that we can detect phenotypically, so they've been ignored, or they have been so detrimental to the plant, they've been eliminated by natural selection. So at the end, I cannot find any obvious reason why transformation and any induced mutations from it should either increase or decrease the likelihood of activation of any long dormant pathway that might happen to be there. So I'm going to finish by thanking my colleagues from ILSI, IFBiC Task Force 9. A lot of what I presented was formulated together with them. So with that, thanks a million.